Hi guys, I thought I should make some videos while the holidays are on, make it a bit easier to catch up with later. So here we are, we've got chapter 11, and chapter 11 is all about these different molecular tools and techniques available to us when we start playing around with genes and DNA. So we've got restricted enzymes, Electrophoresis, the Southern Blot technique, named after a guy called Southern, Polymerase chain reaction, DNA sequencing, solving crime, of course, so some uses of this, determining who's the father, and the ability maybe to clone genes. So, I'm sort of in the way down there. I'm with that picture, and I should grab myself out of the way. I drag myself up. Oh, I've lost the thing. I'll have to do one. Restriction enzymes are used to cut DNA, so they're like the scissors, if you like, when we're talking about cutting and pasting genes. And so each particular restriction enzyme, or endonuclease, has what's known as a recognition sequence. So it's a series of nucleotides that repeat. So they cut in a specific location, and they always cut in precisely the same location, so that's really important. They're capable of producing two types of end, and in their diagram there you can see the first one. This is essentially a blunt end. So it's been cut, but we've ended up with a straight edge there. These are sticky ends. So you've got some DNA hanging over there, some DNA hanging over there, and you can glue those together again much more easily than you can glue together a blunt end. So essentially we don't look for restriction enzymes which make blunt ends because they're not as valuable, but clearly those that make nice long sticky ends make life a lot easier. Excuse me. Restriction enzymes actually exist naturally. They are a construction of bacteria, and bacteria use them to destroy invading viruses. So they break up the DNA of viruses. So it's a protective mechanism for bacteria, which we've been able to make use of really carefully, cleverly, to cut up is the DNA we want to cut up. Excuse me. So the very, very first one was actually found in Escherichia coli, E. coli. And so it's actually called, eventively enough, E. coli, R1. So echo for E. coli, R for restriction enzyme, number 1. And so you can often see that with each species. It's might be an R1 or a 1 for the first one found in that particular species. And of course, species will have many. So echo R1 is a commonly used one. ALU, I don't know how you pronounce that. Is the first restriction enzyme that was isolated from the Atrobacteria luteus. And we'll see why we work with BAMH3 and all sorts of things. Excuse me. So restriction enzymes, we know how to name them, we know where they've come from, and they allow us to cut DNA at a particular site. Of course, having cut them, you want to put them back together again. And so the glue is the same glue we use, that we talked about already in DNA synthesis and in mitosis. It's ligase. So ligase can then be used again to put together two pieces of the DNA you've cut apart with restriction enzymes. It simply recognises the need to stick things back together again. And it's an enzyme to help put those hydrogen bonds back. And again, naturally occurring, so all living things have ligase, so ligase is easy to find and get hold of. Probably enough about ligase, really. We will run a gel in class, if you haven't done one already. Normally we would have gone to CSIRO and used their wonderful facilities, but they no longer exist for students. So a gel is a really simple way of taking some pieces of DNA you've cut up with restriction enzymes and finding out how long they are. And the length, of course, becomes important, becomes a diagnostic tool. So we get our sample of DNA. We might use one or more restriction enzymes to chop it up. And then each of those pieces is loaded in a well. And the wells are just down here. So we get a piece of DNA and we load those into these wells. And then we run the gel. So we put electricity through it. And these pieces of DNA will move down the gel, leaving bands behind them. So DNA has a slightly positive charge. That's a mark on your exam right there. And being slightly positive, it is repelled by, slightly negative charge. That's a mark right there, the negative charge. Being slightly negative, they're repelled by this negative end and attracted by the positive end. So this whole gel here is sitting in a solution. That solution's ionic and allows for charge to run through it. So we attach the negative end of our terminal here, positive end there, and run a charge through the solution. Because the DNA bits are charged, they move away from the negative end to likes repel towards the positive end, and unlikes attract. Law of electrostatics, isn't it? Something like that. And of course, they also move different speeds. So the bigger the strand, the slower it's going to move. So these are our wells just here, and these ones here we're seeing are going to be larger, and these ones down here are going to be smaller because they're going to move faster. And of course, if you leave it running, it runs out the other end. So you might get a question about, we've got a blank piece of gel. What happened? Well, one of two things happened. The gel was left running too long, and all the DNA has finally worked its way to the end and fallen into the solution, or it was wired the wrong way. So that was the plus end here, and that was the negative end here. Then all the DNA you load in your wells would quite quickly go out the other end. That can be an exam question that's been used in the past. That's wanted you to understand how gel electrophoresis works. And you'll see lots of gel examples in past exam papers. Edward Blot used this technique, or used these abilities, to develop a thing he called the Southern Blot. Well, has been called the Southern Blot technique in honour of his work. So, if you've got a large lot of DNA to work with, so for example you're trying to do a whole genome, you can use this to find specific genes. We'll try and work our way through here. So we've got our DNA molecule. We've used a restriction enzyme or more than one to break it all up. We've put that into an agarose gel, electrophoresis, and run that. And those fragments, of course, will move at different places to different size. We transferred that then to nitrocellulose filter. So there's the gel, there's the filter paper, and we get these lovely bands of marks along the filter paper. And then, if we've hybridized with reactive labels, the X-ray film would show us which ones have actually taken up our hybrid with the DNA probe on it. And so it's a simple technique for finding pieces of DNA that you're particularly interested in. And we'll talk more about, I've just done that through there, I've done it already, haven't I? One of the interesting things was, in the early days with Southern Blot, it was quite common to use radioactive probes. So this is a piece of DNA that causes a radioactive reaction. Of course, lots of people got sick doing that because radioactivity can cause cancers and things. So it's more common now to use a probe with a fluorescing gene. And so you get fluorescence rather than radioactivity, which is not a bad idea, is it? I best watch the time, haven't I? 8 minutes? Ooh, rain down. Reverse transcriptase is an interesting process which can be used in some applications. So it's an enzyme which actually makes a copy from messenger RNA. So we've got the messenger RNA, but of course remember this doesn't contain the introns, it contains the exons. So you can go back to the DNA that would have created it. Now, interesting enough, when people have used this, they've actually found that they can't then create a protein from the DNA they've made. So there's something about those introns that have been removed that are really important to the process of making the protein synthesis that we probably don't understand yet. I certainly haven't read anything that explains it. Maybe I'm just behind my reading. That has appeared in the exam from time to time, but it's a pretty rare, uncommon thing, so I'd sort of throw it in there. The other one that I really love on the exam is PCR, and PCR is such an easy thing. It's a photocopier, really, in many ways. So this is quite common if you've only got a small sample from a crime scene, for instance, or you've found a an ancient bone of a human being in a cave somewhere, like a Red Deer cave people or a Denisovan, and you've extracted a little bit of DNA, but to get more is going to destroy the fossil. So you take a small piece of DNA and you can amplify it. You can put it in this machine, PCR process, and make lots of copies really fast. It's very, very important in crime, and I'll talk about more in class when we talk this through with more time. So the PCR simply disassociates the two strands, template from complementary, adds in a whole lot of nucleotides, creates copies of each of them, and then just keeps on doing it over and over again, disassociating and reassociating. There's a whole lot of detail in that slide. We'll go to that slide in more detail in class. But essentially, you've taken a small sample, and you've course it copied many, many times. It was originally done by hand, and you had to have a whole lot of hot water baths at different temperatures, which meant you had to keep on adding in different enzymes, polymerases, because, of course, they all work at different temperatures. This is where our third polymerase turns up, TAC polymerase. So Thermus aquaticus, the bacteria which describes a thermophile, likes really hot water, has been used to extract its polymerase, and we can use TAC polymerase in this process that allows us just to run the gel. So you set up basically a PCR machine now looks like that little box there, and you add your TAC polymerase to it, and it just keeps on working because, of course, the temperature changes are well within its range. So nice little discovery, that one. We'll talk more about that in class. More of this comes what they have now. Oh well. Stage 1, denaturation. You have to heat it up to about 95 degrees Celsius, and that breaks the H bonds. We put our primers in, and these are then attached to the DNA, the template or the complementary, and we cool to about 50 degrees Celsius for that to occur, and all the nucleotide bases put themselves back on. You raise the temperature to 72 degrees Celsius, and those new strands are completed, and then you just go over and over again. So it's going up and down the whole time, and the TAC polymerase copes really nicely with that. And these sort of things are used to allow us to sequence DNA, because we really want to get an idea of a person's DNA sequence. With the genome, the whole idea was to get an idea of which nucleotide follows which nucleotide on every chromosome, and then which part of those create genes that actually code for something in our current state. A lot of our DNA, of course, codes for genes that we no longer make. They've been switched off, but relate back to our evolutionary development. So gel electrophoresis is often used, and the intent is just to take out samples of DNA and we code them. And it's often done manually, so this is often called a Sanger method, and uses lots of gels. And you get lots of small samples, and you sort of get to know where things are. And you're using restriction enzymes which allow you to cut things. You can use probes that you know the sequence of, and you build them yourself, and it just takes hours of manipulation. And of course, when they first started doing a genome project, there was a concern that it would take many, many years. But being able to automate it, which they were able to do, allows them to produce, when there's a small section of chromosome 1 on a human, the full map of human genes much more quickly than they thought they would. So they've been able to build a machine to do what we used to do manually. Well, 1.5 minutes. What do we use it for? Use it to solve crime. You know about that. You watch TV. And we'll talk about this in class, using probes to find things. We use it to determine paternity, because we have these wonderful little segments called VNTRs and STRs that repeat within family groups, and they're quite easy to find. So we can quickly determine whether you're the son or daughter of a particular person quite quickly. And again, I'll go over those in more detail in class. And of course, the big thing is gene cloning. Can we clone things? Certainly, we can recombine things, and we'll talk a lot about the recombination in class. And we can clone genes. We can make copies of genes, and then use them for things like making insulin for diabetics. I think I just squeeze in. Oh, yeah, we'll do it. We'll have to do this in class because I'm about to run out of time. But we obviously use this in plant changes too. A lot of this in agriculture and horticulture. And this is going to keep going for me, isn't it? Lots of medicines. Again, we'll finish off in class because we're going to run out of time here. Okay, that's chapter 11. We'll meet again soon for chapter 12.